Good morning, students. Today we will be discussing about a little bit more details on hypersensitivity. As discussed in the last class, Introduction to Hypersensitivity, we have studied what is the fundamental basis of allergy or sensitization or overreaction. Excuse me. We have seen 4 types of hypersensitivity reaction, basically based on, for example, what kind of immune response is elicited. For example, from the 1, 2, 3, it's antibody-mediated immunity, antibody-mediated immune response is involved in this hypersensitivity reaction. In type 4, it is cell-mediated in terms of humoral and cell-mediated. We will talk cell-mediated in tomorrow's class, but we already discussed that it is involved with the T helper cells, and then it causes cell-mediated cytotoxicity, and transplant rejection is also one of the mechanisms. In coming on to 1, 2, 3, it all antibody-mediated, but fundamentally it's an antibody-antigen interaction. But individual type changes with in terms of pathophysiology, what is the mechanism involved, what is the response in terms of the hypersensitivity given the time, and what is the consequence of in terms of the pathology, in terms of outward disease symptoms. In terms of mechanism, what happens here, what are the types of examples here that we have discussed briefly. So today we will take a little bit more detail in type 1, type 2 and type 3. And again, as discussed in the beginning of the class, I will put you a kind of video lectures where each mechanism they will discuss in brief, and then we will rehearse it one more time and to go into other aspects, whatever discussed in the terms of syllabus. They want to know us a little bit more on allergy, hypersensitivity reaction, types 1, 2, 3, 4. And in addition to not knowing the fundamentals and different mechanism of the 4, they want to know in terms of what are the symptoms, what is the immunodiagnosis you can do in terms of each of the reaction. So we'll briefly talk about this. And again, this is a descriptive information. You might have to make notes again. I will give you a kind of a medical terminology a little bit and what the diagnosis can be used. But overall, it is a common diagnostic technique. We will see that it can be used for all types of reactions. We will see what is the basis of this testing mechanism. And further, we will talk next week on this aspect. So coming on to this kind of, you may get confused once you close the tab or close the textbook. There is a mnemonic. You can remember 1, 2, 3, 4 based on A, B, C, D. A is the first one, allergy, anaphylaxis, atopy. Based on the allergen is involved here in the first one, immediate and fast response. Some of the symptoms are anaphylaxis, another one called atopy, we will see in today's class. And the third one, it involves an immune complex, not only an antigen antibody in single, so immune complexes are involved. So that is part of the ABCD mnemonic, you can remember that. And although 1, 2, 3 involves antibody antigen mechanism, direct antibody was involved in everything. But the second one, hypersensitivity reaction, it uses or it implements fundamentally all the conceptual defense mechanism, how an antibody works, coming from complement cascade, opsonization, inflammation, all will be used in type 2. So it is a classical example of antibody function. So the 2 is called as B. And the 4 is delayed based on the fast. 1 is fast, 4 is delayed based on that. That's a mnemonic, you can easily remember that. Excuse me. So again, as discussed, each mechanism varies. We will take in depth into 1, 2, 3 today. Before that, I would like to brush a little bit more on fundamentals again. If you understand this well, then you will understand most of the mechanism, whatever explained in the visuals easily. So as suggested, given an antibody, we have studied in the lecture 3 different aspects, properties of the antibody, what are the binds, what is the structure, what is the light and heavy chain, what is constant and variable region, and also glycoproteins involved here. So there is variable terminology given the recombination events of the VDJ recombination, given the antigen antibody binding. But specifically, if you look FAB and FC, so FAB is a fragment, that antigen binding fragment, that variable fragment here, that is described as FAB here. And FC is a fragment crystallizable. So actually it's not the constant fragment. They are saying the C coming because when you try to get these protein molecules separate, FAB and FC, try to characterize them further using biochemical techniques, they could be able to see that this fragment, FC fragment, was able to crystallize quickly. It able to, in a given condition or technical condition, it was crystallizing quickly. So that is called as FC crystallizable, not the constant region. So why I'm talking about the FC region here? So this is interacting with the antigen on the upward here, antigen binding site comes from this side. But we have been FC region also a lot of functional importance in antigen antibody complex and functional implementation, because this portion acts as a ligand. It acts as a ligand, and it can interact with many cells which have a specific receptor for this, called as FC is the ligand, FCR is the receptor. Why we are talking the receptor and ligand here? Because cell signaling is most fundamental and important for immunology, because each cell communicates with the other cell. There are partners, the ligand coming from the cell, signal sending cell, and the receptor in the receiving cell, the signal perception process, intracellular transduction in the cytoplasm, then a cellular response that may come in terms of the nucleus, a specific protein can be produced. In cell-to-cell communication, sending cell will send the ligands, or it may have a ligand as a soluble ligand, antibodies floating, or it may have a ligand attached to a specific B lymphocyte here. It may be attached directly to the antibody. It may be soluble per se, free floating, or may be attached to the plasma cell. So when the ligand interacts with the target cell, the receptor, the target cell may be a neutrophil, maybe a mast cell, or maybe a basophil that may have a specific receptor specific to the antibody. They can communicate. There is a response can elicit, but if there is no receptor, it may not communicate properly. So given that scenario, if you superimpose the same cellular communication, cell signaling in terms of antigen-antibody interaction, how a mechanism of immune response happens. So this is the FC region of the antibody. The pathogen is already recognized by the antibody antigen recognizing site, FAB. This is the FC region. And then this is the receptor on the effector cell. The effector cell may be mast cell, or it may be natural killer cell, it may be some other cell, immune cell. They have a specific receptors on their body. They can recognize specific antibody based on the FC region. There will be a complementarity here to the pathogen, so that there is a mechanism can happen. So if you understand this, most of the lecture will become very easy, because they will be talking about much of this FC and FCR interaction here, FC receptor interaction. So again, I'm emphasizing today I will be using a kind of a graphical lecture with some multimedia videos. I would like to reiterate that I'm not selling this video to get some money. I'm just using it for educational purposes. So I would like to credit the further lecture to the medical teaching resource called AMBOSS. They are the one makes learning for medicine students easy. They have made very conceptual problems very easy with graphics. So we will take one at a time. Now, basically, I will briefly introduce to type 1, the type 1 hypersensitivity reaction. I will show you the graphic here briefly. I will explain. Then we will go in terms of the video, what they will talking about. So basically, they are saying that in type 1, immediate hypersensitivity reaction, the allergen is the antigen. It may bind to specific to the mast cell or basophils where there is a specific antibody. Only specific type of antibody is there, IgE. So IgE antibody is attached to the mast cell already. It already has a receptor on the mast cell, the red one, FCR. This recognition makes a antigen and FC and FCR binding here, and the allergen binds here. So there is a cross-linking mechanism. There is a recognition process, upward, downward recognition. So all systems are ready, set. They have the communication signals ready, network. Then they need to go in terms of that. They will tell the mast cell to degranulate based on the pollen or allergen coming here. Degranulation happens. This releases inflammatory mediator, mainly histamines. They will do specific action. So how they will do that, we will see in the mechanism. I will briefly stop this share, and then I will share the video lecture. So 1 second. Then we go to share, go to the video. So share with the computer sound. We are going now to type 1 hypersensitivity. So this is the link I will provide. You can go and read it in further understanding also. Type 1 hypersensitivity. Any issues with the video or recording?
Immediate response, type 1 hypersensitivity reaction. A hypersensitivity reaction is when the body responds to an otherwise harmless substance which is normally ignored. In this case, the body recognizes the substance and triggers an inflammatory response. The usually harmless substance is called an allergen. The inflammatory response is colloquially referred to as an allergic reaction. An immediate allergic reaction is a type 1 hypersensitivity reaction and requires pre-sensitization. During sensitization, the body's immune system initially comes into contact with the allergen. In tissue, the allergen binds to receptors present on B cells. Part of the antigen is transferred to an MHC molecule and presented on the B cell surface. When the receptor of a T helper cell binds to the complex, the T helper cell is activated and releases cytokines. These cytokines activate the B cell and initiate its maturation into a plasma cell. The plasma cell produces and releases IgE antibodies. These antibodies have two variable regions which enable binding to the specific antigen, here, the allergen. With its constant region, the IgE antibodies dock onto a surface receptor on mast cells which are now able to respond when re-exposed to the same allergen. The individual is now sensitized to the allergen. Upon re-exposure, the allergen binds to the IgE receptors, cross-linking adjacent receptors. This leads to mast cell activation, inducing the release of vesicles filled with inflammatory mediators. This process is termed degranulation. The three most important inflammatory mediators released are histamine, prostaglandin D2, and heparin. There are various routes in how an individual can be exposed, for example, through inhalation or skin contact. Depending on the affected tissue, the released mediators can cause bronchoconstriction, leading to respiratory distress, vasodilation, reducing blood pressure and leading to edema formation, and local inflammation with heat, redness, and itching. One of the special features of type 1 hypersensitivity reactions is that once sensitized, the response to the allergen is immediate and can occur within seconds or minutes of exposure. Other types of hypersensitivity reactions are slower and can develop after hours to days.
Okay, hope there was no problem with the audio. So you were able to easily follow it up. Let's keep this point.
Yes, sir.
Okay,
Yes,
great.
sir.
Great. So we are stuck with the PowerPoint. We have to start again, sending it, share. So any queries there in terms of the understanding? One thing I wanted to say in that slide is that because when they are saying that the B cell was initially sensitizing phase, they talked about MHC molecule. So that there is a MHC molecule comes. So they talked about a specific MHC molecule that interacts with the T cell, because when we were talking about MHC molecule, we spoken MHC class 1 and class 2. We specifically told that class 1 is in general to all nucleated cell, including B cell, red blood cell, whatever. They have MHC1 molecules in their surfaces. And the class 2 MHC molecules specifically to antigen presenting cell that are interacting with the T cells. So that's why don't need to get confused in the sensitization phase that the T cell communicates with the specific MHC molecule on the B lymphocyte, and then it triggers the cytokine production that introduces the generation of the clonal population through plasma cell. That is a clonal propagation theory. That is very important theory again. Okay. Now the mechanism is clear here. So we will simply now, this is I made one more slide getting from the website that you can go back to the image Again, you can read the text. They explain the pathophysiology simply here. Then if given an essay, you can write about all 4 types, or given one type of immunological hypersensitivity, you can talk this mechanism. Oh, so that is whatever they are spoken here: degranulation, everything, histamine release. And in terms of what are the symptoms, what in the visually symptoms the doctor or physician can recognize, it will be increased smooth muscle contraction, vasodilation, vascular permeability, lot of rhinitis, cramping. There's a hypoxia condition similar to the whatever we discussed in anaphylactic shock last class, and also extra capillary blood, erythemia, extravasation, and fluid shift into the interstitial space, causing edema and pulmonary edema. The bumps are swelling of the body, you see, the lips also. And another visual condition is called pruritus. So the pruritus in medical terminology is nothing but an urge to itch. For example, if a mosquito bites, you get a bump and you cannot resist, you need to keep itching it. Like that, when immunological reaction happens, you get a lot of itches on the body. There is a lot of, it's nothing but pruritus is itching or urge to itch. So, and then eosinophilia is a condition where eosinophil neutrophils are kind of malfunctioning. That's one more condition. So these conditions may be common to most of the allergy reaction. Make a note of it. So one more specific condition, one more they talk in terms of type 1 is called as another condition called atopy. Atopy is nothing but a genetic predisposition to producing IgE antibodies against certain harmless environmental allergens. They call pollen, mites, molds, certain foods. They're saying that some people, for example, of the 40 class today, 10 people are more allergic than 30 people. That 10 people are not allergic at all because they are not, their genetics are strong. Their lineage in terms of their parents, grandparents, no one had an allergic symptoms. There is no genetic basis in terms from the genome. So based on the external condition, they may encounter. So possible common symptoms are asthma, allergic rhinitis, allergic conjunctivitis, food allergies. They may have some genetic basis or some environmental basis, a condition of atopy. Urticaria is nothing but whatever you are seeing here, these skin patches. Each patch, they may differ in the size, they may differ in the deposition or the position in the body. This is a visual inspection of, they are saying that in this area, there is a heightened response from the immune cells. You may find in an immunological test, a lot of mast cells are here. They may give pinkish or reddish pink and a swelling, and these swells are called wheals diameter. They call wheals. They may change as a small pimple. They may change as a big rash, depending on the severity of the symptom. So another angioedema is also mast cell in subcutaneous tissue. Then another anaphylaxis we have seen in the last class. These 4 may be also symptoms of coming from type 1 hypersensitivity reaction. So given the part of the syllabus, they wanted to see how do you see the symptoms. Symptoms are this, nothing but these are the symptoms. Immunodiagnosis. How do you diagnose hypersensitivity, whether it's type 1, type 2 or type 4? How do you do that? So basically, any allergic reaction, a kind of physician or a trained person, practitioner, he has an expertise in this field, and he will do with a procedure, usually on the hand. He will, with the sterile needle, he will put the drops of a suspected allergen. He has a chemical composition that is identical to the allergen, maybe pollen, maybe snake venom or something else, or maybe a kind of coming from cat or dog hair. They may have the chemical compound, or basically histamine. Histamine is the one produced from mast cells. So they will drop it on the hand and they will see that given a timeline whether an illicit response of immune response are there and a reaction is happening, cross reaction, and they will measure the size and length of the wheel, shape of the wheel, whatever the bumps here, and they will say that this person is actually allergic to histamine, basically saying that he may be allergic to all the pollen molecules because mast cells are there. He may be allergic, not allergic to sheep wool because the negative control is here. He is using the bar up and down. Both spots are put here. Negative control, no reaction. Histamine, positive reaction. Sheep wool, he is not allergic. But horse, dog, cat, feather, many things is allergic. Even the pollen, the dose is common. he may vary in terms of the response. Smaller wheel is small response, bigger wheel is a bigger response. That's the way you will design, that you will test the allergic reaction. If you go into the fundamental of the test, the diagnostics, you call it in two terms. One term is called in vivo skin testing. Another one is called in vitro testing. Basically, you are going the test in the test tube in the laboratory diagnosis, or you're specifically here, you're giving it the patient in a living condition, in the live, you're testing in the skin. So basically, whatever I've discussed in the previous slide, that is the mechanism explained here. General principle is that allergens given, you see for reaction, takes 20 minutes, size of the wheels, but it is another terms are called skin prick test, scratch test, intradermal test, based on the whatever phenomena of the skin is involved. And then one more important thing is that why you want to do in vitro testing. You can go with the in vivo testing, right? Why you want again in vitro testing? Because in a given scenario, a patient X may be more hyper or more responsive compared to patient Y. Whatever given the small dose, maybe he has sensitized before, so he may go into anaphylactic shock, or eventually he may die, that you don't know. That's why people sometimes prefer, given the test anaphylactic shock, they have the medical procedure ready to protect the patient, giving antihistamines, corticosteroid, anticorticosteroid. They can save the patient, but if the situation doesn't exist, they will simply go for in vitro testing. For example, we know that there are more histamines, there is more IgE antibodies in given type 1 hypersensitivity. So you take the blood test in blood circulatory system, you measure that how much allergen in terms of specific to IgE, allergen specific IgE is there. You can diagnose it, you can measure it, you can do that. The more of this allergen specific IgE is there, then he is more susceptible to that allergen or response, giving the response. And another way you can do the mast cell activation, histamines, or you may test that in a given system, mast cells are hyperactive. You can look for this enzyme tryptase in the serum, just like looking for a normal blood diagnosis test you can do for tryptase, then you can find that it is also a type or hypersensitivity susceptible. That's a way you can do that. That's a diagnostic. It may be more common to all the methods. We will see in further. I think it's common to all of them, but it changes in terms of type 2 and type 3. You go for IgM and IgG, here IgE, and in another case, you go for T cells. Okay, that's the case. Now we will go back into the video of type 2 hypersensitivity. It is called cytotoxic. It involves all the classical antigen-antibody interaction, opsonization, complement activation, and in terms of natural killer cell, macrophages also. We will simply go into the graphics now, look into the video. It beautifully explains this process. Then given the time, we will go into type 3 or not, we will see. Okay, stop share the new link, then we will go with that, share. Then I might close this, minimize this. Do I minimize that? Okay, minimize, close this. We are going into type 2 now here. Type 1 is first and fast. You can read given the time, little bit more on that. Type 2 we will talk now.
Antibody-dependent cell-mediated cytotoxicity, type 2 hypersensitivity reaction. During a hypersensitivity reaction, the body can also respond to its own components that are usually ignored. In a type 2 reaction, this response is caused by the presence of certain antibodies. Under normal conditions, antibodies are produced during an adaptive immune response to fight off pathogens. In type 2 hypersensitivity, the antigen is located on the cell surface or is part of the connective tissue. This leads to an antibody-mediated immune response to the body's own cells. The antibodies initiate a cascade of events that can lead to cell lysis, phagocytosis, or apoptosis. Therefore, a type 2 hypersensitivity reaction is also called an antibody-dependent cell-mediated cytotoxic response.
So, okay, they're talking about antibody involvement, and also cells are involved, immune cells are involved. That's why antibodies cell-mediated cytotoxicity. These cells will produce such cytokines or cytotoxin material that will cause the problem. And also, most importantly, you should remember that there is no Fc receptor here per se in this given cell. The Fc receptors may be coming from this cell with the antibody binding, but this specific binding to the cell is to the self antigens. For example, red blood cells or some other membrane cells, important immune cells, there may be self antigen. The binding to the self antigen only causes this kind of hypersensitivity reaction. For example, in blood transfusion also it is important. RBC is having a self antigen. And for example, in a given scenario of autoimmune diseases, this is important. Autoimmune diseases, type 2 hypersensitive will come.
Let's take a more detailed look at the mechanisms involved. Antibody binding to the antigen primarily activates the complement system. This system comprises specific plasma proteins that act as a cascade, mediating further activation of immune cells and heightening the intensity of the response. The complement cascade is triggered by a complement protein that binds to the antigen-antibody complex on the cell. This leads to the activation of further complement proteins through cleavage of small fragments. These fragments are chemotactic, meaning they attract immune cells such as neutrophils, which bind to the antibody via Fc receptors present on the surface. These Fc receptors can detect and bind to the Fc portion of an antibody. After binding, neutrophils generate aggressive substances such as radical oxygen species. These substances cause cellular damage, inflammation, and eventually cell death. In addition, activated complement proteins can bind to the target cell surface, marking it for a process called opsonization. These marked cells can be recognized by phagocytes, notably macrophages, which also use Fc receptors to bind to the antibody. After binding, macrophages start to engulf the target cell and break it down. Furthermore, activation of the complement system also leads to the formation of a membrane attack complex, in short, MAC. This complex can insert into the plasma membrane, resulting in an influx of extracellular fluid. As a result, the cell swells and eventually lyses. Bound IgG antibodies can also be recognized by natural killer cells, often referred to as NK cells. These lymphocytes also possess Fc receptors on their surface, which bind to the antibody, leading to natural killer cell activation. The natural killer cell releases perforin, a protein similar to the membrane attack complex that inserts into the plasma membrane and causes target cell lysis. Natural killer cells also release granzymes, a group of proteins that initiate apoptosis. In some diseases caused by type 2 hypersensitivity reactions, antibodies may be directed towards cell surface receptors, thereby disrupting normal cell function. There are two cases that can be distinguished. First, the antibody blocks the receptor and prevents signals from being transmitted. Second, the antibody binds to a receptor, activating it in the process and acts as a transmitter. Both can have severe physiological consequences.
So you see, in this scenario, you're not looking for FCR. So see, the flipped antibody is flipped here. So in this cell, it is not looking for FCR, but what it is looking is that rather than with antigen binding site, it is binding to the specific cells, disruption of normal cell function. Whatever the receptor present here, that may be a cell cycle division receptor or cancer defending a receptor. So suddenly what happens, the antibody somehow finds a complementarity with this receptor rather than antigen, or maybe complementarity specific to this receptor. It goes and binds. The binding can have two consequences. Worst consequence is that the normal ligand that comes to binds to this receptor is inhibited through this antibody. This is really important in terms of when you are looking for monoclonal antibodies. We will talk further. So it is blocking the normal function. This ligand is not attaching to the receptor. This function is inhibited in the first case. In the second case, there is no interruption. Basically, it is acting as a ligand and it is eliciting a subsequent second signaling pathway, and it is causing some other function rather than the normal function. So this is typically different from normal pattern of FCR binding or antigen binding. And this one we use it as an advantage to define some monoclonal, polyclonal tools that can be used to inhibit some viral diseases or cancer also. We will talk further in that.
As we've just seen, type 2 hypersensitivity reactions usually cause cell death. This can either be complement-mediated or complement-independent. However, for most type 2 hypersensitivity reactions, multiple mechanisms contribute to the autoimmune reaction to varying degrees. It's important to note here that cell death via lysis most enhances inflammation through the release of cell components.
Okay, does it make sense? You are able to follow it? Hello?
Yes, sir.
Yes, sir.
Yeah. So we will now look into the slides, and then you can go back to the video in your free time again. You can look into that. We have less than 3 minutes, so I think we will stop with type 2. I will go to the slides, and then we will start type 3 and type 4 tomorrow. We will start with the sharing the slides. Okay, hang now. Sharing it gets confused. Okay. So it's nothing but here you are seeing type 2 hypersensitivity, whatever we have discussed, IgM, IgG, the mechanism. You can look into that again, binding FC receptors. One thing is complement dependent reaction, saying that complement cascade is involved here in most of the mechanism. It's because complements will bind in opsonization, they may cause direct lysis, they may cause indirectly through bringing a specific phagocyte based on the FCR and complement interaction. Complement independent thing, direct with the FCR, natural killer cell will come and it may cause apoptosis and another change in the signaling and cellular function. You can read it bit more detail here. The text is provided again from AMBOSS. You can go through that. You can make a note of it. Coming in terms of diagnosis and all of this mechanism, it is as simple as whatever discussed in type 1 hypersensitivity. Same reactions can be used, but here you are testing the diagnosis for in terms of IgM and IgG molecule. Maybe some complement proteins you may try to detect because enhanced complement activation will come up here. It may not be there in the type 1. So that is the way it goes on. I think I don't want to go to the type 3 and then we don't have enough time to share the video and all. Let's meet tomorrow and then talk about type 3 and type 4 graft reaction. And type 3 is again, it's type 3 is nothing but you're talking simply in terms of another process of giving a response to an antibiotic or a response to antivenom, because you might have been bitten by a snake and you need to expose, you need to administer immediately antivenom. But your body, if it responds adversely to antivenom, then it will not allow the antivenom to work, bind to the venom, so it will break it. At the same time, antibiotics you are trying to vaccinate, and if your body is thinking that vaccination is also a problem, it may respond through type 3 hypersensitivity, and basically it is not doing the benefit for you. If you do antivenom don't work, basically the person will die, because rather than protecting, the immune system is working in the reverse direction. So we will take that tomorrow's class, type 3 and type 4, and then we will stop the lecture for this week and leave next week. I will take seniors. After that, we will resume with interleukins and interferon signaling. Okay, then have a nice day. So we will meet tomorrow and then 1 week later. Okay, I will end the meeting. Take care. Okay.